So homeostasis is maintaining a stable internal and internal balance. Basically, homeostasis means that everything is even on either side, or the inside of a cell or an organism is going to have exactly what it needs, and so it can maintain that amount. The cell membrane and the plasma membrane is the number one thing that controls the homeostasis within a cell. And it's made up of a couple things, and it has some properties. First, it is a phospholipid bilayer. Phospholipids are these little squiggly things, and we're going to talk about those in class. But you can see they have little tails with a head, and the tails are facing inside of the cell, and the heads are on the outsides of the membrane. It also contains these proteins and carbohydrates. It's called the fluid mosaic model because it looks like a mosaic painting where lots of pieces fit together. So it's fluid because it has lipids, and the lipids is another word for fat, like oils. So it helps keep the cell membrane nice and fluid. The number one property of the plasma membrane is that it is selectively permeable. Selective permeability means that some things can go in while other things can stay out. And the cell membrane keeps the integrity of the cell by not letting too much of one thing get in, or the wrong things like drugs, toxins, chemicals. Anything that would harm the cell should be protected by the cell membrane. Now, if you look here, there's a channel protein that always is open. Then you have a gated protein that can be closed or open. And then you have a transport protein where the cell goes, where whatever needs to get into the cell will travel through there. All of these things are going to allow certain things in and out. It just depends on the size. Anything really, really small is only going to fit through here, the lipid bilayer. The water would go through there. Okay, so let's look at the phospholipid. The phospholipid bilayer is this part right here, where water is on both sides. Now, the thing about the phospholipid is that it has polar head and non-polar tail, which means that this part, the fatty acid chains, are scared of water. They're what they call hydrophobic. Okay, hydro meaning water, phobic meaning scared of. And then we have the head, which is hydrophilic. Hydrophilic means water loving. So these things can be exposed to water while these will not, meaning the water will pass directly through the cell membrane, but it should not stay within the cell membrane. Okay, we talked about selective permeability. If a membrane is selectively permeable, only certain things can pass through. Things that can pass across the cell membrane are things that have non-polar molecules, so alcohols and steroids. Small molecules like water can pass through. Things that actively pass, actively means it takes energy, passively means no energy used. Actively passing across is going to be polar molecules like ions and larger molecules. The larger molecules would be like sugars or any large particles that the cell needs to provide. Types of transport, we've got active, which requires energy, and passive, no energy. Think about if someone is sleepy, are they considered active or passive? Most likely you should have said they're passive, meaning they're not doing a whole lot. They're not utilizing a lot of energy. They're like trying to save it. Someone's active, they're moving around, and it requires a lot of energy. Today we're going to talk only about passive transport. Part 2 of this video is going to be about active transport, which you guys will probably watch tomorrow. Now, passive transport can be broken up into two areas, diffusion, and diffusion has osmosis and facilitated diffusion. Passive transport is the movement of materials that does not require energy. We talked about that when we looked at the chart. Okay, let's look at what's happening here. We've got ink molecules, and they go from here to here. So basically what's happening is there's a high concentration of dye here, or ink, and eventually, over time, it spreads out. Over back here is the real life version of it. So the ink comes to the middle. Eventually, over time, it evens out. So molecules are traveling from an area of high concentration to areas of low concentration, making it an even mixture. Diffusion is passive movement from an area of high concentration to low of molecules. So molecules that are moving, spreading out from areas where there's too much of them to areas where there's less is diffusion. Okay, concentration. Concentration is the amount of substance in a given area. So if we were saying dense concentration would be like a lot of students in one class. It's very concentrated. So in looking at concentration, this is high concentration, meaning there's a lot of substance in one area, and this would be low concentration, spread out. All right, in diffusion, this is what happens. So things move from an area of high concentration to low. That's passive transport, and that is with concentration gradient. So imagine the class is full and it's time to leave. Everyone's going to fall out of the classroom to an area where there are less people, low concentration. And this is what happens with molecules of sugar and water. So here is a permeable membrane, a membrane like a sort of plastic. You will be working with some dialysis tubing. And what happens is you have a high concentration of sugar, low concentration of sugar here. Probably even amounts of water. After diffusion, those sugar molecules go through the selectively permeable membrane and even out so that you got pretty much the same amount of sugar over here than over here. Things don't want to be crowded in one area. They really want to spread out. Some things that are going to affect diffusion is concentration. Diffusion always goes from high concentration to low, too much to less. Temperature. So temperature is going to speed up diffusion. Think of sugar that's in cold water. It doesn't really want to dissolve, but if you add heat, then that sugar is going to actually dissolve, and that's why we put sugar in hot tea and not in cold tea. And another thing is molecular size. So the bigger the molecule, the longer diffusion takes, meaning like in order for something to really spread out evenly, the molecule should probably be a lot smaller. The larger it is, the longer it's going to take to diffuse. Osmosis is like diffusion, but it is the diffusion of water. So water through a membrane, water moving from an area of high to low concentration. So let's look back at that sugar solution. So we've got sugar, we've got the membrane, and we've got water molecules. Okay, less water here, more water here, less water over here. Can you see that? All right, so through osmosis, what happens is the water over here decided too much water. I need to be somewhere where there's less water. So it moved to an area of low concentration. And so now there's even amounts of water particles on each side. This is just another look at osmosis. So we've got glucose on this side. The membrane is here. The next one, you see the arrows. So water is moving in, a little bit of glucose moving the other direction. And if you look at the test tube size, that's what's happening. Then on this side, that water is going to build up, and that's how you get osmotic pressure. And osmotic pressure is the force that's produced by water moving into a cell. We're going to talk about a couple cases where osmotic pressure became too much for a person, and they actually their cells could not withhold that much water. There is such thing as drinking too much water, and we'll talk about that in class. So, solutions. We're going to talk about a couple of solutions that show how water can move. A hypotonic solution, water moves from the solution into the cell, and that causes the cell to swell. That rhymes. Or it can actually break, and that would be considered lysis. A hypertonic solution, the water moves out of the cell, causing the cell to shrink. In an isotonic solution, no movement of water in or out of the cell. No net movement. So that means that water is going both directions in the same capacity. So with the types of solutions, here we go. A red blood cell looks like this normally. It has a nice little curve and dent on the inside. So that's how we know it's a healthy solution that it's in. An isotonic solution would have water going in and water going out at the same amount. In a hypotonic solution, that would be less, if let's say salt to water content. So hypotonic would have little to no salt. And what happens is the water from outside of the cell recognizes that there's space inside of the cell, and it rushes in, and little water is left. So this could cause it to blow up so much that it would lyse, meaning pop. And in a hypertonic solution, if you look at the color of the background, it's highly concentrated with something. Let's say it's salt. So that means that there's not much water on the outside of the cell and a lot of water on the inside. Well, the water on the inside of the cell is going to be like, hey, that seems like that's a place I want to be, less water. So the water will leave the cell and little water is going to be going in. So that's what happens when cells are exposed to a hypertonic solution. It's almost like a dehydration. All right, why doesn't the plant cell undergo lysis? That's a question we're going to answer in class, but I want you guys to think about that and bring that to me. Why won't a plant cell undergo lysis? I'll give you a hint. It has to do with the cell wall. Okay, so in class tomorrow, you're going to draw what the cells will look like in the different solutions. So that's just a preview of what we're going to do in class, probably as a warm-up. All right, and then here's this cartoon. So it says, watch out, he's harmed, watch out, he's armed, and the slug has some salt. So why are the other slugs really concerned about this? And so you can tell me about that also tomorrow in class. Okay, facilitated diffusion is when molecules are helped by the proteins that are embedded in the cell membrane. So these particles are trying to get into the cell. What it does is it goes into this protein. The protein makes sure that it fits, and then it opens so that that molecule can get into the cell. It's actually used by pressure, so that any sort of concentration gradient is going to help push those things through these proteins. Facilitated diffusion is just basically what I just explained. Certain things can get through the phospholipid bilayer, like water, oxygen carbon dioxide. And then some things have to go through the channel. Larger things like some sugars. What go through facilitated transport, which means it would have to check through a protein. The protein, this one, flips around and it lets that sugar out into the cell.